Hello everyone, in this session we are going to study single cell protein and tissue culture. Okay, single cell protein में याद रखना है कि 3 to 10 kg of the grains are required to produce 1 kg of meat by animal farming. More than 25% of the human population is suffering from hunger and malnutrition. There is a blue-green algae called spirulina that can be grown easily on materials like wastewater from potato processing plants containing starch and straws, molasses, animal manure and even sewage to produce large quantities, and can serve as food rich in protein, minerals, fats, carbohydrates, vitamins. मतलब सब कुछ तो आ गया, कार्बोहाइड्रेट, प्रोटीन, फैट, तीनों चीज़ें आ गई, और मिनरल्स भी और विटामिन्स भी. And such utilization also reduces environmental pollution. Certain bacterial species जैसे कि मिथाइलोफिलस, मिथाइलोट्रोफिस, ये मतलब पूरा एक ही नाम है. So there's मतलब ये बैक्टीरिया है that because of its high rate of biomass production and growth can be expected to produce 25 tons of protein. The fact that edible mushrooms are eaten by many people in large-scale mushrooms culture is a growing industry makes it believable that the microscopic fungi too would become acceptable as food. Then मैं एक बात, हाँ ये क्वेश्चन था कि सोर्स ऑफ सिंगल सेल प्रोटीन यूनिसेलुलर और मल्टीसेलुलर ऑर्गेनिज्म दोनों ही हो सकते हैं. सिंगल सेल प्रोटीन का मतलब ये नहीं है कि यूनिसेलुलर ही होगा हमेशा, उसका सोर्स मल्टीसेलुलर भी हो सकता है. अच्छा, सिंगल सेल प्रोटीन के कुछ सोर्सेज हैं. एग्रोबैक्टीरियम इज नॉट अ सोर्स ऑफ सिंगल सेल प्रोटीन. कौन कौन से हैं? क्लोरेला, सच्चारमाइसिस सेरेविसी एंड मिथाइलोफिलस मिथाइलोट्रोफिस. ओके, एक टिश्यू कल्चर टॉपिक है, तो इसमें क्या याद रखना है आपको कि during 1950s whole plants could be regenerated from explants, that is any part of the plant taken out and grown in a test tube under sterile conditions in specific nutrient media. So this capacity to generate a whole plant from any cell or explant is called as totipotency. Medium must provide a carbon source जैसे sucrose and also inorganic salts. क्या क्या होना चाहिए आपका जो कार्बन सोर्स हो जिसपे कि हम ये जो अपने एक्सप्लांट्स को ग्रो करवा सकें, तो उसमें कार्बन सोर्स लाइक सुक्रोज, इनऑर्गेनिक सॉल्ट्स, विटामिन्स, अमीनो एसिड्स, ग्रोथ रेगुलेटर्स जैसे ऑक्सीजन, साइटोकाइनेंस. So this method of producing thousands of plants through tissue culture is known as micro propagation. और हाँ, इसमें से जितने भी plants बनते हैं they are genetically identical to the original plant which they are grown from. That is their soma clones. Soma clones मतलब कि genetically identical to the original plant को हम soma clones बोलते हैं. Many important food plants जैसे कि tomato, banana, apple, they have been produced on a commercial scale by this method. Then another important application of this method is the recovery of healthy plants from diseased plants. So even if the plant is infected with the virus, the meristem apical and axillary is free of the viruses. So hence one can remove the meristem and grow it in vitro to produce virus-free plants. And scientists have even succeeded in culturing meristems of banana, sugarcane and potato. ओके, एक ये था इसमें कि scientists they have even isolated single cells from plants and after digesting their cell walls have been able to isolate the naked protoplast surrounded by plasma membrane. Isolated protoplast from two different varieties of plants each having a desirable character can be fused to get the hybrid protoplast which can be further grown to form a new plant. These hybrids are known as somatic hybrids, while the process is known as somatic hybridization. The fusing of two protoplasts of good and different qualities. This is what you have to remember. बस इसका somatic hybridization का एक example होता है पोमैटो, ठीक है, जो कि टोमेटो एंड पोटैटो के combining hybridization से बनता है. ये दो टॉपिक्स में बस इतना ही है, अब बाकी हम नेक्स्ट वाले में करेंगे. बाय.